So just to recap a few ideas of that video, in your exam, you will need to know what is a sex cell, and remember, you need to remember what is a homunculus. And these ideas of the Russian doll, some of them will appear in your readings. So remember that they thought that there were tiny humans in the gametes, and these are actually pictures, the one that he showed, of papers of the 17th century. And these ideas of the Russian doll, that there will be a human inside of a human, inside of a human, inside of a human. This is actually a hypothesis that there will be embryos inside of the embryo. So remember those few things. This name is also very important. This idea of the Russian dolls will be very important for your test. Okay, other ideas that I want that you remember. First one is that as a result of the conception, it was only just a transfer of a mini human, and the only thing that it was happening in a woman is that there was an expansion of the mini human. That was the only thing that it was happening, and finally, this means that there was no code. There was no code according to these ideas, and there was only miniaturization. In addition, go to your readings and check these ideas, the relationship between the religion and the preformation that are discussed there, because they could also be in the test, and we can discuss those in class. So another idea that is important to remember is this cell theory, that until microscopes were invented, this debate was not solved, and even there were two types of preformists. So one considered that the homunculus was in the male, and the other one considered that the homunculus was in the female. So remember, we will talk at the end of the class about the invention of the cell theory thanks to microscopes, and these ideas of preformism was divided into two. One that they considered that the homunculus was in the sperm, and the other one that the homunculus was only in the female gametes. However, as microscopes start getting better and better, they start realizing that actually the gametes, they never look like that. They never resemble to small adults. So for the test, just remember, I will ask a definition like this. So in epigenesis, you go from an early stage that go from an organized stage to a late stage that is organized, and this stepwise fashion is very important. So in other words, this new structure like the eyes come from all the structures, and this is important. So we have pre-existing ones that are forming the new ones. There is not only expansion and growth. Remember that the person who was in charge of proposing this idea in the beginning was Aristotle, as we discussed before. And this is also important why they were not able to accept these ideas, because there was no mechanism. And if you take with me later a class in developmental biology, I explain in an entire semester all the mechanisms that we know so far. So we can go from an embryo that is disorganized into a human, and we still have many questions. After centuries of discoveries, there is still a lot of questions. So it was much easier to explain everything in terms of growth, because there was a mechanism, and this is why it was so difficult to accept in the beginning. So these five ideas maybe could be very useful for you for the test. And there are so many important scientists in each as a part of this debate. And ironically, I always talk about the importance of experiments. In this case, the early experiments, the microscope, were not very good, so they support the wrong ideas. While as we didn't have any evidence in favor of this, they reject these ideas in the beginning. And remember, these ones were false positive because the microscope have a very low resolution, and you never will find a homunculus in the gamete cells. So those ideas were wrong, and now we are moving more into this direction. Later in the class, I will talk about how the microscope were invented, how from this that doesn't look like a microscope at all, people start seeing more how cells look like. And as I said in the beginning, they look like this. But will be a topic for a future class in which we will discuss more the type of different microscopes. Now, in this last section of the lecture, I'm going to talk about the present and also about the future of this debate. And although we might think that epigenesis completely won, there are still a lot of things about this. And actually, the preformation, right now we have different type of preformation. So now we think, for example, that to some degree DNA have all the instructions, and if we have the DNA, we have all the rules and all the instruction of this. And so there is still this debate. It has new approaches and new ways to explore these ideas. So let's talk about that. So one of the problems that the homunculus idea had, for example, was to explain phenomena like the regeneration of a limb. This will be impossible to explain through a homunculus. And also it's very difficult to explain how this infinite regression will take place. So these are few problems that we still have with the homunculus idea, in addition that of the microscope, that we know that they don't exist. But these were one of the key indications in the beginning of that these were wrong. So there is some degrees of truth in both. So for example, we know that the homunculus is wrong. However, not all the ideas about the epigenesis are correct. So for example, we say that they go from an organized state to an organized state, and this is not always completely true. So let me show you an example. So as I said, neither is totally correct. So both have some degrees of truth, and let me show you an example. For example, with epigenesis, definitely we see a gradual process. And this is an example of the development of an invertebrate. And here you can see that from cells that look like an embryo, that are you form as a sphere, you end up with a completely different organism. However, although this gradual progression is true, when we went and tried to see if in the beginning the expression it was very disorganized. So for example, the gene expression in fruit fly, we thought that it would be very messy in the beginning, and we found that this is wrong. Actually, when we went and studied the gene expression in embryos of fruit fly, is very organized. So here you see colors, and each color represents a different gene. And as you see, they look pretty organized even from the beginning. So this idea that they go from an organized state to an unorganized state is not always true. Even from the beginning, they can be organized. And we have shown that in multiple cases, and here I show you a case of the gene expression in fruit flies. So remember, this is not always true. And now I'm going to show you something. So maybe, although again the homunculus is wrong, there are some degrees of preformation that are actually right. So for example, we actually have instructions that to some degree influence. So we are to some degree preformed in the DNA. One, it could be good. However, if we think about, we take it again to the extreme, we might say that we are genetically determined. And this is not completely true. So there is a big influence from our genes, but they don't determine everything in our body. And in class, we will discuss some of those ideas. Is there free will? Is there completely genetic determinism? And how we find the boundary between both. When, in which sense we are determined, and which case we have free will. And an extreme view, the genes determine your life, actually we found that it is not true. And these ideas come from these early findings that one gene can produce one phenotypic trait and another gene can produce another phenotypic trait. So there was a simple relationship between genotype and phenotype. And these ideas were in the 70s and 80s and even before that. However, we know that these occur only in a few cases. So, and now we know that there is a complex relationship between genotype and phenotype, and let me explain that. So, definitely we have sometimes gene can be responsible for one trait. However, this is not always the case. Most of the cases is very different. Yeah, we find that one trait can depend on multiple genes. So, for example, here if you see gene 2 is affecting trait 2, trait 3, and trait 4. And the trait 2 is also affecting not only for gene 2, but also for gene 3. And we have found that actually gene expression is very important, that the dynamics of this process is fundamental. So, for example, here, if you see, I show the line intensity, very different. So, the degree of expression is very important. So, in trait 5, you have a lot of expression of this red gene, while in the trait 3, you have a small expression. And also, the spatial and temporal patterns are very important. So, there is not a simple idea, and it's more complex as we thought. So this complete preformation is actually not true. And in class we will discuss this idea of predestination, this genetic determinism between the idea of free will, and we can start thinking about mechanisms and in which case we have one or in which cases we have the other one. And this is all for today. I hope that you have fun today, and hopefully in class we can discuss this in more detail. Have a good day. Bye.